So greetings to everyone on this call today. I welcome you all to today's webinar on emerging electronic sensing platform for life science applications. Just a few housekeeping notes. Today's session is being recorded and all the participants are muted by default. You would have to unmute yourself to directly ask any question, or you could also use the chat window. My name is Priya Mangannavar. I'm a micro nano electronics technologies technical staff at CMC Microsystems. Today, I'll be providing a brief introduction to CMC Microsystems and the electronic sensor platform. Professor Ebrahim Ghafar-Zadeh then will present about his research and how they have used this sensing platform for their biosensing applications. So let's get started with knowing a bit about CMC. CMC Microsystems is a not-for-profit organization with a mission to lower barriers for technology adoption. In our mission, CMC delivers micro-nano innovation capabilities across Canada and, broadly speaking, deal with technologies that will enable Canada's digital economy of the future. We typically work in the domain of microelectronics, photonics, IoT, MEMS, quantum computing, and many more. We are actually independent of the application. It really doesn't matter what drives you, whether it's advanced manufacturing, clean tech, agriculture, or oral health sciences. We are here to provide you the tools to help you carry out your research and accelerate your development. We also have partners all over the world. We are part of many of the different communities. So if there's any information that you require on specific markets or you want connection in other countries, we'd be very happy to help you with that. So the infrastructure and services we provide is divided into three main categories: CAD, which delivers design tools, platforms, and compute infrastructure to design and develop micro and nanosystem prototypes. We deliver these capabilities either through the cloud infrastructure or on the user's desktop. Next is Fab. We provide Fab services, which has multi-project wafer services, package and assembly, fabrication and travel assistance to manufacture prototypes in commercial or university-based labs. Under lab services, we provide short-term access to development platforms, test equipments to validate and emulate test prototypes. We also provide services for emerging processes and products. The process we are talking about today, the electronic sensor platform, is one such example. This platform was first demonstrated at an university lab, and now it has grown to become available to users across Canada and beyond. So at its core, the electronic sensor platform consists of a silicon junction field effect transistor, and it is delivered without an upper gate. This allows for integration of a functional material, which would act as an upper gate to modulate the channel conductivity. Now, this flexibility in depositing the functional material of your choice makes this platform suitable for a plethora of applications. The first application for which this platform was developed was for optical or photo detection using colloidal quantum dots, with those as the functional material. Since then, this platform has diversified into several other applications, especially in the field of biosensing. In today's webinar, we have Professor Ebrahim Ghafar-Zadeh from the Department of Electrical Engineering and Computer Science, York University, talking about how he has made use of this platform for biosensor applications. Professor Ebrahim has completed his PhD at Polytechnique Montreal and then obtained his postdoc at McGill University and University of California, Berkeley. He then joined as an associate professor at York University and is the director of Biologically Inspired Sensors and Actuators Research Lab. His research is aimed at exploring novel integrated sensors and actuators for life science applications. I would now like to invite him for his presentation. I would like to hand over the presentation to you, Professor.
Thank you. Thank you very much for being here. Thanks CMC for organizing this webinar and for introduction. So today I want to talk about the Open-Gate Junction Field Effect Transistor. That has been the fabrication of this. Actually, the sensors have been facilitated by CMC, and we have used this sensors for various applications. Today I will share with you a number of the applications. So field effect transistors have attracted the attentions of researchers for a variety of applications for life science. So two main groups of the applications can be considered biosensors and chemical sensors. So the biosensors have been developed using FET for a variety of applications, including COVID-19 detections, for detections of the cancers and other type of the, let's say, diseases. And the chemical sensors as well, for example, one of the main application, pH measurements, and also for measuring different type of chemical solvents and solutions. Here, this diagram shows how the applications of the FET have been promoted over the last few years. And in addition to the large number of the papers that have recently been published, the commercializations of FET-based sensors have been also started over the last few years. So one of the applications that I will show actually the DNA sequencing that has been, they use the standard technology for that purpose. Okay, let's start with ISFET, ion selective field effect transistors, which is one of the FET technologies. So this structure indeed include drain, source, and the external gate, as you see. And indeed, the presence of the chemicals, charges, biological materials may increase the charge density between the drain and source. And the variation of these charge density results in the variation of the current that can be measured. So in addition to these electronic or electrical parts of these structures for biosensors, for chemical sensors, we need a sample holder in order to assure the sample only be held on the top of the sensors and doesn't reach the bonding pad and doesn't actually cause any short circuits problem. So among different materials, of course, silicon is one of the materials that have been used for the fabrications of the FET devices, including ISFET. So here, this diagram illustrates the mechanism of the sensors. Just I'm repeating that the presence of the charge may result in the changing of the conductivity, and the change of the conductivity in the end will result in the change of the current. So the variation of this current should be detected in order to detect the, let's say, for example, the target molecules or cell. So the accuracy of the systems depends on many different actually factors. Of course, the interface circuits or the readout systems, and also the sensitivity of the device. So the goal in FET FET sensors is to increase the sensitivity. So here in this actual presentation, I want to show one of the very sensitive devices that we have developed using Open-Gate. So among various standard technologies, the CMOS, Complementary Metal-Oxide-Semiconductors, have been widely used for developing actually different type of the sensors, including ISFET. So in this technology, they use the top most metal layers and the bottom layers in order to create a kind of extended gate ISFET. This device the CMOS has been used for the fabrication of the large number of ISFET on the single chip. one of the very important applications, DNA sequencing. As you see, this is the simplified diagram of this structures, that the presence of the bead coated with DNAs can actually result in the change of the pH in the presence of the AGCT molecules, and the variation of the pH can be detected using the sensor. So this device has already been commercialized. Ion Torrent actually has commercialized this device. This is one of the actual version of this device that include more than 165 million wells. So the advantage of the CMOS for a large number of the sensors is very actual, has been actually shown very clearly in this specific product. So the fact here is the CMOS still is considered one of the actual important platform, but the main issue that we have with the CMOS sensors is the silicon area. So indeed, using actually a CMOS limit us to very, very small chips, and of course that can be used for specific applications. So the idea is how we can actually increase the sensing area that Open-Gate actually address this challenge. So again, advantage of the standard technologies in general is to develop actually a large number of the devices in order to decrease the cost. ISFET itself has the general, actually, I can say, problems that is the external gates. So external gates can be developed on the topmost metal layer, for example, if we have, let's say, CMOS or even Open-JFET, but the fact here is the voltage, or actually accordingly the charge density generated by the external gate might be actually affected by the ionic solution. So this is why this can be also considered as another problem. Open-JFET also addressed this issue by using a backgate that I will show you in the next slide. So in addition to the silicon, other type of the materials also have been promoted in the recent year. For example, carbon nanotube can be used as a semiconductor between the drain and source, and also, for example, the graphene can be used as another alternative. Indeed, these two materials or other type of the organic and nanomaterials also are being actually, different type of the sensor are being developed, and these materials have also demonstrated very good applications. However, still the scalability of using this type of the materials is one of the topic in the research. Therefore, still I can say the silicon is one of the materials that can be adopted. For example, any technology using the silicon for developing the FET technologies, FET sensors, can be adopted with the available platform technologies. So another one point that I want to raise here is that the type of also the curves that has been used, for example, for these type of the sensors. As I show you actually in the previous slide, that we try actually to reach the currents to the saturation actually phase. That area can be used for sensing. However, in the new materials, the sensing area is rely on the variation of the conductivities, and in the first phase of these characterization curves. Okay, so now the point here is, in order to address the issue that I mentioned for the external gates, and the solution, which is the Open-Gate, is Open-Gate Junction FET transistors as a solution, can be seen in these structures. So this structure simply shows the drain source and an open window on the top. So that means there is no any metals, there is no any other, let's say, materials, and the let's say biomaterials, chemical materials can access to the surface of the device. So in theory, you can see there is no any material, but in practice, actually, a native silicon oxides can be actually observed should be observed actually on the top of this channel. So again, the most important advantage can be considered the foundry process can be used for this type of the actual silicon-based material, specifically Open-Gate that has been recently facilitated by the CMC. It allows us to design actually our devices just using software, and also the device can be fabricated and we can actually use it for different applications. This is what we have done over the last couple of years. So this shows actually give us a better idea of the structures. So this is the detail of the structures can be seen here. The backgate. And interesting actual point is this, that the distance between like a backgate and the channel, because the channel actually is created on the top actually of the wafers, and the backgate is exactly in the back of the device. Despite this actually distance, this thickness, the system actually, the device shows very well actually functionality and very well sensitivity. So again, as I said, we have focused on developing actually different type of the devices, ion OG-JFET devices. Let's start with one of the devices that we have used, and we demonstrated the functionality of the device and applicability for different life science application. So in this chip, you can see 7 different sensors, 7 different transistors. These transistors have been, the source of this transistor have been connected to each other, and the drains are open to be connected to voltage, individual actual different voltages. So that means each one of the sensors, it can be used as a single sensor. So this device, as you see, is large. The chip is large, so allow us to have a large pad. So we can access to this actually pad without using any wire bonding. So indeed, here in this specific for this specific sensors, we have used a spring load in order to connect it, and that makes actually the development of the make it very easy for us to make a prototype for testing and for measurement purposes. So, and yes, this device, so before actually the development of the device, we used the console for actually studying the functionality of the device, and also finding the optimum length and width for this actually for each channel of the transistors. So here you can see a diagram, simulation results, the variation of the current versus the voltage drain source. So as you see, the variation of the VGS has results in changing the current, and indeed this can actually tell us that the voltage higher than 3 volts can be used for sensing purposes between 3 and 4. And assuming that we want to use this area as, for example, for sensing purposes. As I mentioned before, we prefer actually use this part, this area. However, this area also is informative, because if, for example, by changing the voltage, still we can see the variation of the conductivities between drain and source. So another simulation results here shows the variation of the current drain source versus the voltage across the gate source for different voltage drain sources. So this is one of the important actual curves that if you are interested using the Open-JFET, indeed the voltage gate source, the variation of the voltage gate source can be considered actually an electrically actuations of the channel. And indeed, the effect of the voltage gate source on changing the drain source resistance or conductivity is very similar to the effect of the charges that we introduce from the top of the device, because those charges change the charge density. Similarly, the gate source can play the same role. So this is why these curves can give us a better idea of the sensitivity and also the functionality of the device when it is exposed to the biological or chemical materials, include charges. So the first task after developing the device is the characterizations of the device. So here it shows actually one of the standard actually measurement setups that we have in the lab, which include the semiconductor characterization systems and the probe station. So this device, these two actual instruments are used in order to first of all reach to the bonding pads and using these probes, and then connecting them to the semiconductor characterization system. So also we can use here actually we can use the same setup in order to introduce, for example, sample as you see here. However, this liquid sample actually is using the liquid actually droplet, for example, on the top of which is a little risky. Why? Because it may reach to the bonding pad and also make a kind of short circuit. So for this purpose, we use this device mainly for dry characterization, and we suggest this used for dry characterization. So the one of the big disadvantages of this instruments is that there is actually, these are expensive and many of the labs don't have actually this set of the equipments. Okay, for the wet characterizations, in order to avoid the issue I mentioned before, we may need to address this issue by developing microfluidics. So here you can see a custom-made board that allow the connection of the chip on the surface of the PCB and also connecting those devices to the system, to the characterization, semiconductor characterization system. And also we develop a microfluidics. These microfluidics allow the injection of the solution into the channel and reaching to the sensing side on the top of the sensors. So this microfluidic device also allow us to make any kind of changing of the, for example, the pH continuously and so on. Okay, so we demonstrated the advantage of this device for different applications. So first application is the pH measurement. So we can change the pH using the same setup that I mentioned in order to and use the OG-JFET sensor for measuring the charges, create charge density, and accordingly actually the current of the drain. So as you see, the variation of the pH can show actually a linear change. So important point here is the pH measurement is one of the standard measurement that if you have even any other applications, maybe the pH measurement, it can be actually suggested as the one of the actually basic actually testing procedure, because the variation of the charge on the surface of the, let's say, chip can be changed, and the functionality of your device can be measured. So the presence of the charge, or the, let's say, concentration of the charges can directly change the charge density, and the change of the charge density can show the change of the variation and change of the current between the drain and source. So here also you can see the variation of the shows the repeatability of the sensor. So by changing the pH, we can see the variation of the output of the sensor. and over time. So of course, these times can give us also an idea about the response time of this device. And as you see, the response times is it's about a few minutes. So the fact here is this time is required in order to see the effect of the charges that are accumulated in the capacitance, the layer capacitance created between on the top of the device. And of course, this response time is very depending to the size of the sensor. For example, for the very large sensor, of course, we can see a larger actually response time. So another important application is the DNA detection. So DNA has the detection of the DNA is important for many different applications, as you know. So indeed, for the DNA storage applications, we need to store, let's say, for example, to have a specific volume of the DNA for sensing purpose. Sorry, for DNA storage purpose, or for example, for other applications. Let's say, for example, for the infectious disease detection, indeed, what is detected is the let's say, a specific gene or a specific sequence of the DNA. So what we have demonstrated is the measurement of the DNA concentration. This has been actually shown for the first time. The reason that the Open-JFET was the perfect for this purpose because the top of the device was open. That allows us actually even to increase the sensitivity. So the results shows the nanogram per microliters actually variation of the DNA can easily actually result in the change of the current. So that means we can use actually the Open-JFET for measuring the DNA change. So another point that is important to say again, DNA measurement also can be considered as one of the good actually test procedure in order to verify the functionality of your device. If even you have actually you have developed the device for different application, because the DNA include actually the negative charge. This negative charge can be controlled the volume, the amount of those actually charges by just changing the concentration of the DNA, and this allow you actually to see the characterization, to see actually the functionality of your device and the linearity of the device as well. Okay, in addition to these two applications that I showed, we have also demonstrated the advantage of the Open-JFET for many different applications. Some of the actually published actually results can be seen here. We still working actually on Open-JFET sensor for the other application that will be published and discussed in the future presentations. So we can add, let's say, for examples, we have added the different type of the waters on the surface, ultra pure waters, and also we put the PBS and let's say, for example, because the difference between these waters are the, let's say, concentration of the ions in this type of the, let's say, water. So that could give us actually an idea how actually these device, let's say Open-JFET sensors, can be used for measuring actually the amount of these ions. So also we, in addition to introducing the liquid like the water with the different ions, also we just actually created the very actually thin layers of the liquid on the top of the device, and it was very sensitive because the fact here is, let's say, like breathing air, it's include actually a very small droplet of the of the water, and that can be spread on the surface of the chip, and still the device is very sensitive to show that. In addition to this application, also we have shown the advantage of this device for monitoring the oral neutrophil, oral cells, so the cells that are in saliva. As you know, there are two different types of the cells, epithelial cells and also the the oral neutrophil in the saliva. So we have also shown how the variation of those cells can change the charge density and accordingly the variation of the color. So just two of these results. One here you can see actually the presence of the breathing, let's say, air that is actually make a very thin layer of the liquid on the surface of the chip has results in the change of the current very much significantly. So another diagram shows the variation of the variations or repetition actually, and the variation of the concentration of the oral cells actually in the liquid, in the ultra pure water. And that shows actually that this, for each one of these actually, let's say for example repeated still, we can see the linearity. So indeed the device was developed actually in general, for example, for different application, and we use it for this as well. In order to use, let's say for example, develop a device for cellular monitorings, a specific actually attention should be made for examples for the size of the device, let's say bits, length, and some other geometries. So in addition to that, also the surface of the device should also be considered as another important factor. So so indeed, indirectly, again, as I mentioned before, the pH can be used to give an assessment of the surface charge. And if I want to actually, let's say, just give a summary of the main actually functionality of Open-JFET sensor, I can say the Open-JFET is the best actually for measuring the charge density caused by, let's say, for example, the cells or with the other type of the molecules, and that can actually perfectly be used for the sensing purpose. Okay, so our journey in developing, designing, and also the simulating and fabrications and characterization of the device can be seen in this flowchart. So if you are interested in the developing Open-JFET device, so for you need to specify the application, of course, for different application, the geometry of the device, size of the channel, and so on, number of the band. All of these will be a function of your interested application. Second is the design specification, and then the simulation. Simulation again can be done using the consoles. We have developed a console app that can be used by the members of the CMC. That these can help you actually just use this console app to specify the parameters and so on. That I will show you on a slide about that. So then the layout is fabricated, and the fabrications can be actually done through the CMC. And then most important part is after the most important steps after the fabrications are the packaging, characterization. So the packaging is, for example, one of the packaging idea can be the microfluidic device that you need to develop for a specific application on the top of the chip. And also you may actually develop the rapid prototyping that I will show you one actually of our results also today. So characterizations can be used as standard technology, a standard measurement setup that I showed you, the probe stations and also semiconductor characterization system. Which are expensive. So for this purpose also we have developed, we address this challenge by developing a portable multi-channel FET characterization system, which is much cheaper and can be in access of the CMC members. So here is the console applications. Simply actually you need to set the parameters, run the program. And indeed, in this, you have numbers actually of the parameters that you can control and use these applications. So if there is any question about that later, actually I or one of my actual PhD students who designed this software can answer the question. So here is the characterization system that I mentioned. So in this characterization, so I showed you actually this characterization, let's say setup. That include actually a semiconductor characterization system and also the probe station. As I mentioned, this is expensive, around actually $100,000 to $200,000. So we develop a characterization system that can be used for this purpose. This characterization system, Open-Gate characterization system, include the software, include the hardware system, and also include a package. So indeed, we have already used the actually 3D printing method that we developed in our lab in order to create actually the required actually electrical connections between the chip and device, and also to create a sample holder on the top of the chip. Okay, so let me summarize my talk today. So today I talk about actually different applications of FETs for life science, and also specifically I moved into the detail of the Open-JFET device, one of our devices that we have developed recently, and I showed you actually different applications of that. So in addition to that, I actually show you two of the products that we develop actually beside our research that can be used by our networks, and these are actually the hardware system, the characterization system that include hardware and software, and also the packaging method. Thank you very much. Let me thank my students. First, Abbas Panahi, Hamid Usuli, Tabrizi, Ahmed Roshamper, and Murtaza. And also, I should thank the support, great support of the CMC in all technology, including Open-JFET, support of the MyTechs that support my students, and also the answer. Thank you very much.